Hello, I am Professor Sims, and this video is about adaptive immunity. This is the second of 10 lessons included in my pathogenic microbiology course. If you are currently enrolled in this course, please consult the syllabus and the Moodle site for assignments and other course information. The learning objectives for this unit include distinguishing between primary response, secondary response, humoral immunity, cellular immunity, antigens, epitopes, and different classes of antibodies. We're also going to identify cells that express MHC1 and or MHC2, so that's your major histocompatibility proteins. We're going to describe the structures and cellular location of these molecules. We'll identify antigen-presenting cells, antigen processing and MHC presentation, the process of T cell maturation, thymic selection, and diversity. We'll compare and contrast various classes of subsets of T cells, and we'll talk about production, maturation, and activation of B cells, the structure of B cell receptors and T cell receptors, primary and secondary antibody responses, artificial immunity types, variolation and vaccination, and finally types of vaccines and their advantages and disadvantages. There is a lot in this lesson. The phagocytes of the innate immune system. So we talked about the innate immune system in lesson 1. So these phagocytes of the innate immune system are very good at their jobs, but they're not able to identify all invaders. And for that, what's needed is the adaptive, also known as the acquired immune system. The adaptive immune system responds to foreign antigens more slowly than the innate immune system, but it is far more specific. It orders up custom designed proteins called antibodies to fight invaders. And unlike the innate immune system, the adaptive immune system has memory. So when it encounters the outsider again, it remembers its previous defense and can mount it much more quickly. So as this figure shows here, the leukocytes of the innate immune system, they all have different names, they all have different functions, but they're all involved in rapid response, much like your street cops, your fumigators, firemen, riot police. So these are all called out for emergencies, and so are different types of leukocytes. Lymphocytes work a bit slower, but they're the big guns that get called out in a particularly specialized situation. Lymphocytes are like the special forces. And then your dendritic cells, these are the signal men. These are the ones that are directing traffic. In other words, the dendritic cells form a bridge between the innate and adaptive immune response by presenting antigens to the B and the C, to the B and to the T cells of the adaptive immune system. So that's what's shown here. You have your innate granulocytes and monocytes, and then, so these are the different types of leukocytes. Then you have dendritic cells that help these guys communicate with the lymphocytes over here, the B cells and the T cells. Antigens of a pathogen activate the host's active immunity, and they're differentiated by molecular class, complexity, and by size. Lymphocytes are the heart and soul of the adaptive immune system because they can modify their behavior to adapt to novel kinds of antigens. They do this in two ways: humoral immunity, which is carried out by B cells, and cell-mediated immunity, which is carried out by T cells. Humoral immunity is based on antibodies called immunoglobulins produced by B cells. Each B cell carries many copies of a single antibody on its surface, and antibodies stick to foreign antigens and act as kind of a label that declares that the antigen should be destroyed by phagocytes. Cell-mediated immunity works mostly with two kinds of T cells. You have cytotoxic T cells, which especially combat pathogens by identifying hijacked cells carrying foreign antigens and persuading them to commit suicide. Each individual lymphocyte carries at least one unique receptor that can bind one particular type of antigen. Together, the receptor and the antigen in the body's trillions of lymphocytes possess a mammoth inventory of receptor possibilities equipped to deal with almost any antigen. Up to half of our lymphocytes move through blood circulation and the lymphatic system, and the central hub of the immune system consists of organs, for example, the spleen, the thymus, the tonsils, lymph nodes of the neck, armpits, and groin, and of course blood vessels. First exposure of an antigen stimulates the primary response, and the subsequent exposure stimulates a faster and stronger secondary response. Notice that the secondary response is much faster and provides a much higher concentration of antibody. Epitopes are part of an antigen to which an antibody attaches itself. Epitopes induce specific adaptive immune responses. So that's what these guys here are, epitopes. This entire thing is the antigen. An antigen is a macromolecule that reacts with components of the immune system. And a given antigen may contain several different motifs that are recognized by immune cells. A typical protein antigen has multiple epitopes shown by the ability of 3 different antibodies to bind to different epitopes of the same antigen. And that is shown here in figure 18.4. Antibodies, also known as immunoglobulins, they are Y-shaped glycoproteins with 2 sites for binding antigens. They're involved in complement activation, neutralization, opsonization, agglutination, and antibody-dependent cell-mediated cytotoxicity, or ADDCC. Antibody-dependent cell-mediated cytotoxicity. Remember that antibodies are also called immunoglobulins, and thank goodness there is an abbreviation for immunoglobulins. It's abbreviated IG. Immunoglobulins differ in where they operate in the body and how they trigger reactions from the immune system. There's 5 different kinds of immunoglobulins. So you have IgM. This is the first antibody to respond during infection. It is a B-cell receptor. IgG is the most common type of antibody. IgA is present in mucosal secretions. IgE is stimulated by allergens. It activates basophils and mast cells. And IgD is only about 1% of the proteins in the plasma membrane of B lymphocytes. And it usually is co-expressed with IgM, which is the first antibody that responds. So figure 18.5 is showing the structure of a typical immunoglobulin or a typical antibody. Notice the Y-shaped structure. Antibodies must be capable of enormous structural variation in order to be able to recognize the essentially limitless number of foreign antigens that they might encounter. So an antibody is another one of nature's modular structures. It's a molecule made of 4 peptide chains, 2 heavy chains and 2 light chains, and it's shaped like a Y. The ends of these chains, the tips of the Y, are short variable amino acid sequences. In any one antibody, the variable sequences are identical on both of these tips. And the variable regions have different shapes. So the variable regions here, the variable regions are what give antibodies their ability to stick to just about any antigen, and the rest of the Y is the constant region. Neutralization involves the binding of specific antibodies to antigens found on bacteria, viruses, toxins. This prevents them from attacking and attaching to target cells. Antibodies serve as opsonins, so they coat, right? Opsonization, the coating. They also inhibit infection by tagging pathogens for destruction by macrophages, dendritic cells, and neutrophils. The phagocytic cells use receptors to bind to AGG, opsonize pathogens, and initiate the first step of attachment before phagocytosis. Antibodies, especially IgM antibodies, agglutinate bacteria by binding to epitomes on 2 or more bacteria simultaneously. And when multiple pathogens and antibodies are present, this forms an aggregation, and the binding sites of antibodies bind with separate pathogens. So in this way, you have, these are bacterial invaders, these are IgMs, and you can actually have one IgM that binds to more than one invader at the same time, and then that helps to kind of group them together and kind of reel them in. In this example of antibody-dependent cell-mediated cytotoxicity, the ADCC, Antibodies bind to a large pathogenic cell that's too big for phagocytosis, and then they bind to receptors on the membrane of an NK cell. So this interaction brings the NK cell into proximity where it can kill the pathogen through the release of cytotoxin. In cell-mediated immunity, cytotoxic T cells learn to recognize antigens that are combined with major histocompatibility complex proteins, the MHCs, and they're shown to them by antigen-presenting macrophages and other immune system cells. So remember, the major histocompatibility complex, the MHC, this is a collection of genes that code for glycoprotein molecules expressed on the surface of nucleated cells. MHC1 are found in all nucleated body cells. MHC2 are found on macrophages, dendritic cells, and B cells. The antigen binding cleft of MHC1 is formed by alpha 1 and alpha 2. The antigen binding cleft of MHC2 is formed by alpha 1 and beta 2. So what this figure is trying to show is that there are different classes of major histocompatibility complexes, and what makes them different is that they're in different types of cells, and they have a similar but not quite identical structure. Antigen-presenting cells, which are also known as APCs, ingest pathogens by phagocytosis. They destroy them in phagolysosomes. We saw that in the last lesson. They process antigens. They select epitopes with MHC2 for presentation to T cells. The antigen-presenting cells activate the T cells in active immunity. If a pathogen directly infects the cytoplasm of an APC, processing and presentation of antigens occurs. But if the pathogen does not directly infect the APC, then you have to have cross-penetration. When the antigen presentation and T cell activation used by dendritic cells is not directly infected by the pathogen, phagocytosis of pathogen by presentation on MHC1 rather than This is all very confusing. What it's saying is, in a nutshell, is if an antigen-presenting cell is not directly infected by a pathogen, then there are extra steps involved for that cell to be able to recognize the antigens that are on the pathogen, and then for the cell to activate the T cells. So in other words, it can happen directly, it can happen indirectly, but one way or another, the antigen-presenting cell activates the T cells, which helps to stimulate destruction of the pathogen. I want to pause here to show you guys a video.
Antigens are processed differently, depending on whether they originate within or outside the cell. Proteins produced within the cell, such as viruses or self-proteins, are broken down into fragments. Fragments of foreign proteins are antigens. The antigens are then transported into the rough endoplasmic reticulum. The antigens combine with class I MHC molecules. The combination is then transported to the Golgi apparatus and from there to the plasma membrane. Foreign antigens presented on class I MHC molecules stimulate cell destruction by activated T cells. Self-antigens are not recognized by the T cells and do not stimulate cell destruction. A different process occurs if the antigen originates outside the cell. Phagocytes, such as macrophages, ingest foreign particles, such as viruses and bacteria, by endocytosis. The foreign particles are broken down into fragments within a vesicle. The vesicle containing the foreign fragments fuses with vesicles from the Golgi apparatus containing the class II MHCs, and the two structures combine. The MHC class II antigen complex is transported to the plasma membrane. The displayed MHC class II antigen complex can stimulate other immune system cells to respond to the antigen.
MHC1 molecules function as a messenger, displaying intracellular proteins to T cells and preventing immune response directed on host cells. So your MH1 molecules are helping to get rid of pathogenic cells but are protecting host cells from autoimmunity. When MHC1 presents antigens derived from pathogens, immune response is initiated. A dendritic cell phagocytoses a bacterial cell, brings it into the phagosome. Lysosomes fuse with the phagosome to create the phagolysosome, where antimicrobial chemicals and enzymes degrade. So we saw this in lesson 1. You have the lysosome fuses with the phagosome, which makes the phagolysosome, and then within the phagolysosome is where all of these enzymes and stuff degrade the bacterial cell, and then it is going to be excreted by the cell. Proteases process the bacterial antigens. Most of the antigenic epitomes are selected and presented on the cell's surface in conjunction with MHC2, and the T cells recognize these antigens, and that's what activates the T cells. This figure is showing red bone marrow. Red bone marrow is the site of production and differentiation of formed elements. Again, we discussed this in lesson 1. But the bone marrow is where you get your erythrocytes, the leukocytes, and the platelets. That's going to be in the red bone marrow. Yellow bone marrow is populated with adipose cells. The thymus is bilobed, H-shaped, and it is a glandular protein located just above the heart. It's surrounded by a capsule of connective tissue. The darkly staining cortex and the lighter staining medulla of individual lobes are clearly visible in this micrograph taken from a newborn. So we wanted to show where in the body these things are initiating. Okay, so that's why we're talking about bone marrow and thymus. T cell receptors, TCRs, are similar in structure to immunoglobulins. Remember, immunoglobulins are the antibodies. But they are, thankfully, less complex. TCR are divided into distinct gene segments called variable, diversity, and joining segments. So D, V, D, and J segments. During development of functional TCR in the thymus, genetic rearrangement in a T cell brings together one V alpha segment and one J alpha segment to code for the variable region of the alpha chain. Combinations of these rearrangements of V, D, and J provide enough genetic diversity to produce millions of toll cell receptors with unique epitope-specific variable regions. So let's have a look at some of those different types. So different classes of T cells. You have helper T cells trigger macrophages to go forth and seek invaders. They also produce interleukins. Interleukins are a group of cytokines that lead to rapid creation of new T cells and new B cells. Regulatory T cells oversee that whole system. And then your cytotoxic T cells, we've talked about, but cytotoxic T cells destroy cells that are infected with intracellular pathogens. Activated helper T cells differentiate into one or more distinct subtypes. So your Th1 cells, they stimulate cytotoxic T cells. They produce memory in those cytotoxic T cells. They stimulate macrophages and neutrophils. They stimulate NK cells to kill pathogens more effectively. Th2 cells stimulate B cells, and they differentiate into plasma cells and memory B cells. They direct antibody class switching in B cells. Th17 cells stimulate immunity to specific infections. And then memory helper T cells remember specific pathogens, and they mount a strong and fast secondary response.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
Differentiation is directed by specific cytokine exposure. This illustration depicts the activation of a naive or an unactivated helper T cell by antigen-presenting cell and the subsequent proliferation and differentiation of the activated T cell into 4 subtypes. So these are the guys we were just talking about. This is the Th1 cell, Th2 cell, memory helper T cell. So all of these guys are related to each other and they help to activate each other. They work in conjunction with each other. So just make sure that you know these two tables and who does what. Okay. Cytotoxic T cells target and kill the infected cells. So this figure is showing the activation, this is showing the activation of cytotoxic T cells, and this is done by an antigen-presenting MHC1 molecule on an infected cell. The cytotoxic T cell releases perforin and granzymes that invade infected cells and induce controlled cell death. Controlled cell death is called apoptosis or apoptosis. Superantigens are bacterial or viral proteins that cause nonspecific activation of helper T cells. Superantigens lead to excessive release of cytokines, which is called a cytokine storm, and a systemic, potentially fatal inflammatory response. So what's happening in figure 18.19 here? The macrophage is presenting a foreign epitope that doesn't match the toll cell receptor of the T cell. And because the T cell doesn't recognize the epitope, it's not activated. And then over here, the macrophage is presenting a superantigen that's not recognized by the toll cell receptor of the T cell, but the superantigen is still able to bridge and bind to the MHC2 and toll cell receptor molecules. This is a nonspecific and uncontrolled activation of the T cell, and it's happening because of an excessive release of cytokines that activate other T cells and cause excessive inflammation. B cells, or B lymphocytes, this is a type of white blood cell of the lymphocyte subtype, which functions in the humoral immunity component of the adaptive immune system. And the B sites secrete antibodies. So, like T cells, B cells possess antigen specific receptors with different specificities. Although they rely on T cells for optimum function, B cells can also be activated without the help of T cells. B cell receptors, these are different forms of immunoglobulin D and immunoglobulin M, so different forms of these two types of antibodies that bind to specific antigen epitopes within their antigen binding region. Diversity of antigen binding specificity is created by the genetic rearrangement of V, D, and J segments. So this is just like we saw in T cell receptors. B cell receptors are embedded in the membranes of B cells. The variable regions of all of the receptors on a single cell bind to the same specific antigen. So this, again, looks very, very similar to your antibodies and your T cells, but unlike the T cell receptors, B cell receptors do not require antigen presentation with the major histocompatibility complex because B cells interact with epitopes on free antigens. And while TCRs only recognize protein epitopes, BCRs recognize epitopes associated with proteins, polysaccharides, and lipopolysaccharides. Because protein antigens depend on the activation of T cells, they are called T-dependent antigens. Polysaccharide, lipopolysaccharide, and other non-protein antigens are T-independent antigens because they can activate B cells without the help of T cells at all. T-independent antigens have repeating epitopes that can induce B cell recognition and activation without involvement from T cells. The second signal, such as interaction of toll-like receptors and PAMPs, is also required for activation of the B cell, but once activated, the B cell proliferates and differentiates into antibody-secreting plasma cells. In T cell dependent activation of B cells, the B cell recognizes an antigen, it presents it to its helper T cell that's specific to the same antigen. The helper T cell interacts with antigens presented by the B cells, which activates the T cells and simulates the release of cytokines that activates the B cell. And activation of the B cell triggers proliferation. Whoa, there's a lot going on here. So the short version of this is B cells can be activated by antigens that are on proteins, but if it is activated by antigens on proteins, then it does also need the helper T cells for activation. However, B cells can be activated by antigens that are on non-proteins such as polysaccharides and lipopolysaccharides. And in that case, they do not need the helper T cells to become activated. So that's the last 3 slides in English. I hope that helps. Repeated exposure. So B cells carrying an antibody that recognizes and binds to a specific antigen then go on to divide. They create additional copies of themselves in their successful antibody. And these daughter cells are called plasma and memory cells. Plasma cells tend to die fairly rapidly, but memory cells persist. And the memory cells continue to divide. Now that's important because that has to do with the secondary immune response. The secondary immune responses are not just faster than the original. So this is just like what we saw before with the T cells. Secondary immune responses are much stronger. They're faster. They result in antibodies that bind to an antigen much better. The increased strength is the result of mutation in the variable region of DNA fragment, and they come about by chance. So this is figure 18.23, showing that the repeated exposure to T-dependent antigens results in secondary antibody response, which is faster and more specific compared to the primary response. The second antibody response occurs more quickly and produces antibody levels that are higher and more sustained, and this involves mostly IgG, immunoglobulin G. Okay. Adaptive immunity is divided into natural active, natural passive, artificial passive, and artificial active immunity. So these are the 4 classifications of adaptive immunity. Beyond looking at T cell versus B cell, you're also looking at mechanisms of acquisition. So your natural acquired passive immunity is immunity that is acquired from antibodies that are passed from the mother to the child, either through breast milk or through placenta. The natural acquired active immunity is going to be immunity that's gained through illness and then recovery. So initial exposure and then secondary exposure. Artificial acquired passive immunity is immunity gained through antibodies that are harvested from another person or from another animal. And then artificial acquired active immunity is acquired through vaccines. So let's talk about vaccines. A vaccine is a preparation of a particular antigen that activates the immune system without producing disease. The point of a vaccine is to persuade the immune system to mount a defense to make antibodies against specific pathogens, a defense that will be ready if infection occurs. Vaccines are often administered by injection, but some are given by mouth. Vaccines produce an immune response in the same way that actual infection does. The host identifies the vaccine's antigens as non-self antigens, and it gets rid of them and remembers them in case of future encounters. Not everyone in a population has to be vaccinated in order for a vaccine to be extremely efficient. If a large portion of the population has been vaccinated, pathogens will have a hard time successfully moving from person to person because the percentage of vulnerable people will be small. This is what is called herd immunity. The number of people in a population who must be vaccinated for herd immunity to work varies from disease to disease. Herd immunity is also really important because not all people in a population can be vaccinated. So this here, I want to show you, I got this from theguardian.com, and it is a simulation of what happens during a measles outbreak and how much of a population has to be vaccinated against measles in order for herd immunity to kick in. And it's really fun just to see, and it is slightly different every time you run the simulation, so I think that it would be beneficial for you guys to do this on your own as well. So here it starts. This is the 10% vaccination rate, 30%, 50%, 58.5, 68.9, 74.4, 83.8%, 86%, 90%, 99.7%. So these little red people, the red dots coming in are infected persons, people that are infected with measles. Then you have the blue dots are people that have been vaccinated against the measles. Then the yellow dots are people that have not been vaccinated or can't be vaccinated, so they're totally susceptible. And so the simulation runs for a little while, and the red dots come and jump in, and they get in where they fit in. And depending on the vaccination rate in the population, you have more people that are infected based on how many people are susceptible, how many people are vaccinated, and you can see there's a huge drop-off of people that get infected. And it does not have to be 100% vaccination rate for the herd immunity to really kick in and drastically reduce the amount of people that are infected with measles. So for example, you have, almost 90% of this population is infected, and that's because the 10% of the population was vaccinated. But you see how quickly the number of infected individuals drops off, and this population here doesn't even have 70% vaccinated, but it's way more than 70% of this population that is protected by those vaccinated persons. So that essentially is what we mean when we discuss herd immunity and vaccines. The most common vaccine, in fact, is the MMR that is given to children and protects against regular measles, mumps, and rubella. In most cases, these viral diseases are mild, but that isn't always the case. Rubella causes major birth defects. Measles frequently makes patients suffer complications including pneumonia, encephalitis. People can die from measles. Mumps can cause sterility in males. Possible side effects of vaccination include rash, fever, body aches, but all of these side effects are uncommon. In about 1 in every 100,000 vaccinations with MMR, a child suffers a severe allergic reaction. And despite that, the MMR vaccine has been accused of causing autism, though large epidemiological studies in several parts of the world have disputed this claim, and vaccination rates have dropped sharply. Public health authorities point out that as vaccination rates decrease, measles is on the rise, and very, very quickly. Some fear of an epidemic if things get to the point where the herd immunity breaks down. So in early studies on vaccine, we had variolation. Variolation is an inoculation method that was first used in 10th century China, and it was used to immunize individuals against smallpox. This was done with material taken from a patient in the hopes that a mild but protective infection would result. The procedure was quite risky back then, though, because it could cause the disease that it was intended to prevent. Variolation for smallpox originated in the Far East, and it later spread to Europe and Africa. This figure here, 1825, is showing in a Japanese relief the depiction of a patient that's receiving a smallpox variolation from Agata Shunsaki, who was a doctor from 1748 and lived until 1810. He was a physician that lived from 1748 to 1810. Now, thankfully, we are not using variolation these days. Modern vaccination, it was developed by Edward Jenner. Edward Jenner inoculated patients with infectious material from cowpox lesions and learned how to prevent smallpox in that way. So on the left here, this is a painting of Edward Jenner, and there is a cow and a milkmaid in the background. I don't know why that is relevant exactly, maybe because he was using cowpox. But on the right here is an example of smallpox. These are lesions that were infected with cowpox, and it's caused by a virus closely related to the one that causes smallpox. So what he did was he used inactivated cowpox virus and used that to inoculate a patient, which could prevent them from then contracting a very similar virus, being smallpox. And smallpox is very, very, very deadly virus. So Edward Jenner is a hero of microbiology and vaccine development. So this table here, table 18.3, is showing the classes of vaccine. You have live attenuated and inactivated vaccines. You have vaccines that have whole pathogens that have been weakened, those that have been killed, or those that have been inactivated. So your live attenuated vaccines, examples of this are chickenpox, German measles, mumps, tuberculosis, typhoid, and yellow fever vaccines. The advantages are that they have cellular and humoral immunity. They're long-lasting, and they are easily transmissible. The disadvantages is that they're difficult to store and transport. There is some risk of infection in patients that are already immunocompromised, and there is some risk of reversion, which means that sometimes they don't stick and you have to get more vaccines later in life. Inactivated vaccines, example of these are vaccines for cholera, hepatitis A, influenza, the plague, and rabies. Advantages of these are that they are easy to store and transport. There's virtually no risk of infection. The disadvantages is that there is a weaker humoral immunity and they require higher doses and more boosters. More classifications of vaccines include subunit, toxoid, and conjugate vaccines. These all contain acellular components. So your subunit vaccines, this is your vaccines for anthrax, hepatitis B, influenza, meningitis, papillomavirus, pneumococcal pneumonia, whooping cough. Toxoid vaccines are for botulism, diphtheria, pertussis, tetanus. And finally, the conjugate vaccines are meningitis, streptococcus pneumoniae, and Neisseria meningitidis. So that is the end of lesson 2. I want to thank you guys again for watching. Don't forget to do your reading. Check the description below for more comments or more videos related to the topics that we covered. I know we covered a lot of topics in this lesson. Leave your questions for me in the comments section below.